Hi, we're going to talk about the DNA replication process. Here we're going to describe the process of replication, and we're going to describe the role of the different enzymes that are involved in this DNA replication process. So we're going to try to answer what is DNA replication, what's the molecular process, what are the enzymes, so the machinery that is necessary, and what's the outcome. So pretty much what is DNA replication? The name itself, says it. So when you have a cell that is gonna reproduce, right? You're gonna start with one cell and you're gonna end up with two cells. In order to end up with two identical cells, so we're gonna have the parental cell that ends up in two daughter cells. The two daughter cells are expected to be identical to the parental cell, so that they have to pretty much have twice the amount of whatever happens and is present in the one single cell. And so that's what happens with the double-stranded DNA. We start with one copy and we need to end up with two copies of DNA. So just I'll be using this little string, okay, as a proxy for the double-stranded DNA. We're going to see if that helps us. So how is this DNA replicated? So the first thing that scientists wanted to figure out is by, in what way? Is it that, is it a conservative model where pretty much you have a photocopying machine, right? You start with your double-stranded DNA and then somehow there's a photocopying machine. You maintain this original DNA that you initially had and then made a copy, an identical copy of it. And that's what you have, what happens after the first generation or the first replication. And then, in your second replication, same thing happens, where you're gonna have your original parental DNA that will be photocopied into a newly synthesized double-stranded DNA, and then the one that was of the other daughter cell again will be potentially this one, and the new DNA will be photocopied somehow. Or is it under a semi-conservative model? So here in this case, what we're going to have is that somehow the two strands, right, will be split up, and then each of the strands will end up in one of the daughter cells. So pretty much you are conserving one of the strands, but the other strand is newly synthesized. And this happens at the first replication, and again during the second replication, your original parental strand will be conserved and the other strand will be newly replicated. And for the other daughter cell, you're going to use the other strand and there will be a newly synthesized strand. Or is it a dispersive model where pretty much you're going to have sections of the double-stranded DNA randomly, right, that will be from the original strands somewhere in one of the cells and from the other parts in the other cell, and so same thing here. So in the end, the way that this diagram is drawn, right, pretty much it seems that if we had a way to discriminate between the parental DNA and the daughter cell's DNA, so the newly synthesized DNA, potentially we could, see a difference. And so that's exactly what the scientists did. So Messelson and Stahl, what they did is that they cultured bacteria, which are living organisms that multiply super rapidly. So it was just a way of, and they're very easy to culture, right? So it was mostly a way for them to have a fast going experiment. They're very easy to work with. So they cultured these bacteria in a medium, so in a growing medium, where all the nitrogens were actually heavy isotopes. So any molecules in the cell that has a nitrogen, wink wink DNA, right? You know that the nitrogenous base has a nitrogen, then we'll have this heavy isotope. Then they wash these cells, and now they start culturing them in the normal media with the nitrogen 14 isotope, which is the usual lighter isotope that is not radioactive. And so what happens is that what they observe is that after their first generation, when they extract the DNA, they're going to centrifuge it in a gradient centrifugation. And so what they observe is that there's only one band, right? So from the start, a heavier isotope, right, will be migrating further because it is more dense than a lighter isotope that will remain higher up. If we know that the original strand was composed of a heavier isotope and that the newer strands will be composed of lighter isotopes, right, you would imagine that if you only see one band is because all of the cells of these after this first generation actually have just one single type of DNA, which likely will have a combination of both. So if we go back to this idea here, the only models, right, that would help us in that way are the ones that show a combination of the heavier and the lighter isotope DNAs. And so that's the case for the semi-conservative. You can see that both after the first replication, both of the cells will have DNAs that are heavier and lighter, so kind of middle, and also the dispersive model. However, the conservative model should have shown, right, two bands, one that was very heavy and the other one for the other cells that were kind of lighter. So it seems that we can already get rid of this conservative model. What happens in their second replication? So after the second replication, what they end up with is the usual or original middle band here, which is exactly the same one as they had over there. And then they also end up with another lighter band, right? So less dense. And so what does this suggest relating to our models here? Well, we've had already discovered that the conservative model did not work. Now we were left with these two other models, and what we saw is that there are two bands that showed up, one that was identical to the first replication one, and the other one that was lighter. So the only way that you can explain this is because for each of the daughter cells you will have a semi-conservative replication. So now what enzymes are involved in these replications? So there's going to be a lot of enzymes. You're going to have to be patient, make sure that you visualize what's going on, and that you try to almost represent it. And so when I say represented, that's why I'm going to be using my DNAs right here. So one thing, so I made a knot right here at the end of my string. You should also, if you can, go ahead and try to see what happens. And so what you want to do here is try to see what happens with the DNA. So the DNA is usually a double-stranded, so I don't know if you can see, but this string actually has a double helix here. It's actually a triple helix, but let's forget about that. And so what happens is that in order for the all the enzymes to start coming in and start replicating, you need to have this origin of replication that kind of opens up in the middle of the DNA somewhere, right? So somewhere in the middle of the DNA, you're going to open up here and you're going to start separating, right? And so with that, you're going to see how there is a little kind of bubble that shows up, right? So we're going to try to maintain this bubble. You see how hard it is for me to maintain the bubble. The string wants to go back to its original way of being. And then you can see here these, what we call the replication fork, right? So we're going to have the two strands that are separated and then the rest of the double-stranded DNA here. So that's exactly what's going on right here. You have your origin of replication here. That's where the two strands split up. And then you're going to have a replication fork, which are here. So the idea of the fork is that you have the two strands that are separated. Now, one thing I've mentioned, right, is that in my model, and it's the same thing for the DNA, my string wants to go back to its original form, which is double-stranded. So in order for that to prevent from that from happening, I need to have my single-strand binding proteins. So these binding proteins will pretty much put in, the annoying things that will not allow these two strands to come back together. Now, what happened here when I had to separate my two strands, I had to put in some force there, and that was thanks to the helicase. So the helicase is something that is going to break up the helix. As a general rule, any word that finishes with ASE, ASE, is going to be an enzyme. So it's going to be a protein that has role. So another thing that is going to happen is that as you open up the bubble, right, through this helicase, so you release the winding on this end, but the problem is that on this end you overwind it, and so that's going to be too hard for the DNA. And so we need an enzyme that will come in, hopefully clip one of the two strands, and so it's going to have that strand that is clipped unwind itself, and then it's going to link it back again. So this topoisomerase, what it's going to do is that it's going to change the overwounded structure of the DNA, overwound structure of the DNA, and it's going to bring it back to its original structure, right, where you have a certain helix per nanometer, right, or something like that, so that it's not like overturned. And that's going to be the topoisomerase. It's going to release the overwound DNA by creating a single strand nick, and then releasing that strand, and then again linking back together that nick. We're going to have also the primase. The primase is going to be an enzyme that will synthesize an RNA primer. This RNA primer will be absolutely necessary for the last enzyme that we're going to talk in a second about to start synthesizing the DNA. So the primase synthesizes an RNA primer. What was missing in our picture was the DNA polymerase, which is the major enzyme that synthesizes the new DNA strand on a direction 5' to 3'. And that it does using the RNA primer as a primer. The DNA polymerase uses DNA as a template and can use either DNA or RNA as a primer. In the case of DNA replication, naturally it will be RNA primers. In the case of PCR, which is a technique that we'll be using later, it's going to be DNA primers. But here, in the DNA replication process, the cells produces through the primase an RNA primer. Now, this DNA polymerase will synthesize this new strand of DNA in the direction 5' to 3', and you can see how these can keep extending by adding new nucleotides at each time. The DNA polymerase, the way it's going to be adding the new nucleotides is going to be doing the following. We talked about the orientation of the strands from 5' to 3'. So here we have the template strand from 5' to 3'. You can see here again, the reason it's called 5' is that the phosphate is bound to the 5' carbon of the sugar, and this sugar then has a 3' OH that will bind to the phosphate of the next nucleotide. And so this is the same thing that's going to be happening here in the newly synthesized strand. So the newly synthesized strand has a 5' phosphate, sugar, phosphate, sugar, phosphate, sugar, and the sugar at the 3' OH at the 3' end has a hydroxyl function that will react with the nucleotide triphosphate. And in particular, this will be done thanks to the DNA polymerase that will be able to match a purine with its corresponding pyridine, a purine with its corresponding pyrimidine, or a pyrimidine with its corresponding purine, and will cleave this phosphodiester bond here, which will lead to the release of the pyrophosphate, and which allows to give a lot of energy, and that energy will be used to bind this the OH of the prior sugar to the phosphate of the current nucleotide. And so that's how you're going to start building up your whole DNA strand. As you can see here, again, we end up having a 3' free hydroxyl function for the next nucleotide. So here we have a depiction of what happens in E. coli. So Escherichia coli is a bacterium. Bacteria have circular closed DNA genomes. And so pretty much what happens is that you're going to have an origin of replication, which is a specific sequence in the genome, and that will suggest that the DNA has to be opened up at that location. Now you have replication forks that are opening up on both sides, and now you can start having the replication process happen. And as you can see here, right, the replication process will happen on both sides, like towards both replication forks, and you're going to end up with two DNAs that are semi-conservative, as we mentioned before, and each of them will go onto one daughter cell. In the case of eukaryotic cells, the molecules of DNA, the strings of DNA, are very, very long, and so it wouldn't be possible to have a replication process that happens with only one origin of replication. So there are multiple origins of replication through the chromosomes of these eukaryotic cells. But otherwise, it is a very similar process. They are equivalent. And so you also end up with two daughter DNA molecules. So the first part that I explained, right, seemed pretty straightforward. And it was, I explained it actually on the leading strand. Remember that the synthesis process happens from the 5' end to the 3' end. Okay, so that's also why I usually talk, whenever I talk about a DNA sequence, I always talk from the 5' end to the 3' end. And this is a convention among biologists, molecular biologists, microbiologists, anyone. So what happens here is that when I sequence in that way from the 5' end to the 3, synthesize in that way to the 3' end, it's very easy. It's pretty straightforward. Now so we have a continuous elongation, right, of the strand in that direction. At this point, you should be able to complete replication 1. However, this bubble of replication actually, as you can see, has this leading strand in one way, but the other strand will have will be the lagging strand, and we're going to explain why that is the case. So we talked already about the leading strand, we talked about the helicase, about the single-stranded binding proteins, we talked about the RNA primer that is synthesized through the primase. Once the RNA primer is synthesized, the primase leaves that region. And then we're going to have the DNA polymerase. In some cases, it's the DNA pol 3. We're not going to talk about the different types of DNA polymerases. So we're going to have the DNA polymerase 3 and the sliding clamp, which is, something that accompanies the DNA polymerase. And so as shown here, you have a continuous synthesis of this strand. Now, what happens in the lagging strand? In the lagging strand, it's actually a little bit more complicated because you still have to maintain that 5' to 3' synthesis direction, but the strand is kind of going backwards. So what you need to have is, as soon as the origin of replication opens up a little, a primer needs to be made not too far from the origin of replication so that then the DNA polymerase can start sequencing here. But you need to understand that once the origin of replication opens up, this part here is still kind of bound to the other strand. So there is no space to start putting a primase higher up. So you're going to have this first fragment that will be synthesized, and this is called an Okazaki fragment. And then you're going to have the same thing happens with, once the fork is opened up a little bit more, you're going to have the second Okazaki fragment that will be synthesized. Now, the first fragment will have an opening with the next fragment. And so these will have to be fixed. So another thing that needs to be fixed is actually the fact that this strand here still contains RNA. And this happens also in the leading strand. It's just that it happens only once in the leading strand, because for the leading strand you only need one primer. And so this RNA has to be eliminated and has to be replaced by DNA. And that is the role of the DNA polymerase. So the DNA polymerase will synthesize DNA on a 5' to 3' direction. It will also be able to chew up nucleic acid, in particular RNA, right? So it will be able to chew up RNA. It's also able to chew up DNA, but for the process of replication, it will be chewing up RNA. So it will chew up this primer, and as you can see, it chews it up in the same direction as it synthesizes. So it is able to have this, what we call exonuclease activity. Exo because it starts at an extremity, and it chews up the nucleic acid from that extremity. So it's an exonuclease activity from 5' to 3'. And so finally, we need to have the ligase. So ligase is an enzyme that ligates, that links, okay, that creates that ligation between the two fragments of the same strand that were not yet linked. As we look at the full picture of the replication fork, I want to point out three main activities of the DNA polymerase. We talked about two of them. We talked about the DNA synthesis, the 5' to 3' DNA synthesis of the DNA polymerase, right? So from a 5' end, it starts extending, in this case this Okazaki fragment, but it also did it on this end right here. Then it also has, we talked about it as well, when it chews up the primers that have to be replaced by DNA, that's a 5' to 3' exonuclease activity, right? So same direction as the synthesis. And then a third activity that we haven't talked about is proofreading. So the same way you type, and as you type you read, and as soon as you make a mistake you delete, right? You go backwards to delete all those letters and start retyping properly. The DNA polymerase does the same thing. So it is synthesizing, makes some mistakes, notices that mistake, so now it's going to start chewing up all those nucleotides that it placed previously, so it's going to go backwards. So if it goes backwards, it's going to have a 3' to 5' direction, and it's going to have these exonuclease activity as well, right? So it starts from an extremity, and it chews up those nucleotides. So again, the three activities are 5' to 3' DNA synthesis, 5' to 3' exonuclease activity, where it chews up the primers and it replaces them by DNA, and 3' to 5' exonuclease activity, where it proofreads any mistakes it might have made. And so that's pretty much what happens in the replication fork, right? You're going to have a leading strand where somewhere there was a primer that allowed to initiate this whole leading strand synthesis, and you will also have several primers synthesized by the primase that will allow to synthesize with the DNA polymerase these Okazaki fragments in the lagging strand. So here's another picture. You can pause and look at it. But it's very, very important that you understand every single role of each one of those enzymes and proteins. So take-home messages are that DNA replication is semi-conservative. One strand of the parental cell will end up in each of the daughter cells. The DNA replication requires a lot of enzymes that will initiate DNA replication at the origin of replication, with the two replication forks at each end, and that like that they will be able to replicate both the leading strand and the lagging strand. Each of the enzymes are important, and you must know both their role and their names. And finally, I want to remind you of the three major activities of the DNA polymerase: the 5' to 3' DNA synthesis, the 5' to 3' exonuclease activity, and the 3' to 5' proofreading exonuclease activity.